Okay, so for this chapter, chapter 15, it's going to be divided into two parts. And so the first lecture is going to look at two ways in which proteins are sorted. And then the second part looks at the third way. Okay, so we're just looking at intercellular compartments and protein sorting. So how do the proteins get to the location, the organelles in which they need to? So we want to keep in mind that cells are surrounded by the phospholipid bilayer, so the plasma membrane, but also inside of them, for eukaryotic cells, there's all these organelles too, which are also membrane bound. Okay, so for this lecture, for this chapter, we're really focused on the eukaryotic cell. Microbiology, you'll talk about the prokaryotic cell. So as a microbiologist, it saddens me a little, but there are eukaryotic parasites, so I can get excited about that. Okay, so again, these organelles, whether we're talking about endosomes, parasomes, ribosomes, lysosomes, mitochondria, Golgi apparatus, the ER, both rough and smooth, nucleus, those are all going to be made up of the phospholipids, but they're also going to have proteins, and so we have to make sure that we get those proteins to those different locations. So if we look at the cell and look at the percent of that cell volume in which the organelles take up space, we could build a table that we're looking at here. Okay, and so the cytosol is going to take up the largest amount of the volume, and in that cytosol there's going to be free proteins moving around. But then if we look at the mitochondria within a eukaryotic cell, like the hepatocyte, which is a liver cell, 22% of the cell volume would be taken up by mitochondria. So if we think about it, if we went into a cell and we took out all the mitochondria, we would have 22% of the cell just being taken up by these energy-producing mitochondria. When we look at the ER, we can break that, it's about 15%, but that can be broken down in the rough ER, which are going to have ribosomes attached to it, so 9%. And then the smooth ER would have 6%. Okay, and then we have these other, the nucleus is 6%. Also, I was going to say these other miscellaneous vesicles, organelles, but your perisomes, lysosomes, and endosomes are these specialized vesicles that are going to have membranes, and they're about 1% each. Okay, and so for this table, now we're looking at the total cell membrane and where the different organelles are contributing to the total cell membrane. And so traditionally we think of the plasma membrane, and it must take up so much of the cell. I mean, that's the major membrane that, if you were to ask a student, like, name a membrane, they're going to say cell membrane, and they mean the plasma membrane. But plasma membrane. But when we look at how much of the cell membrane is actually the plasma membrane, it's a very small percentage. And so for liver hepatocytes, it's only 2%. With pancreatic exocrine cell, it's only 5%, right? So it's very important, but there's a lot of other membrane material inside the cell. So before we talk about the other organelles, we want to keep in mind what is a liver hepatocyte and what's its function, and what's a pancreatic exocrine cell and what's its function. Okay, so pancreatic exocrine cell is going to produce proteins and secrete them out, exocrine. Okay, for liver hepatocyte, your liver is meant to detoxify, so there's going to be a lot of detoxification that's going to need to be done. And so if you remember back to like an intro bio class, and where you would have talked about the different organelles and what were their functions, we can start to think about, well, depending on these different cell types, what organelle do we need to be more developed and more of, right? So for pancreatic exocrine cell that's going to be producing lots of protein, what organelle is it going to need more of? Well, protein synthesis is really important for that, right? And so we're going to have to make sure we have a really developed ER, endoplasmic reticulum. And so, especially the rough ER, right? Because that's where you're going to have the ribosomes binding to and allowing those polypeptide chains to move into the ER so then they can get folded and processed and everything. And so for the pancreatic exocrine cell, we see it has 60% of its total cell membrane is just in the rough ER, right? So that is the dominant organelle where it's contributing its cell membrane to, okay? Now, when we look at the liver hepatocyte and look at its ER, you'll notice that the smooth ER is much higher than compared to the pancreatic exocrine cell. So they have 16% of total cell membranes contributed from the smooth ER, and the smooth ER is important in detoxification. And so it would make sense that the cell that's detoxifying has a more developed smooth ER, okay? Its rough ER is at 35%, which is, you know, it's going to be making proteins and such, so it needs to make sure it has lots of rough ER in order to do that. But again, that you can see how the level is very different compared to an exocrine cell, which is, its function is to produce protein and secrete it. Another area you'll notice the difference between these two cells are with the mitochondria. Now you'll notice that the inner membrane for both is significantly higher than the outer membrane. The inner membrane is that, if you remember back to learning about the mitochondria, the inner membrane is the one that's going to kind of weave back and forth. There's going to be these cysteines in which all of these energy reactions are going to occur. The outer membrane is just allowing for the trapping of the hydrogen ions during the electron transport system, okay, or electron transport chain. So in prokaryotes, it's called electron transport system, and eukaryotes, it's called electron transport chain. And so I'm teaching microbiology, I'm so used to it being called system. Okay, but for eukaryotes, chain. So during the electron transport chain, we're going to pump hydrogens out, and they're caught between that space between the outer and inner membrane. And that inner membrane invaginates and goes in and out, so you get more surface area where you can have energy occur, and so hence why the percentage is higher for your inner membrane versus your outer membrane. Liver cell needs to have more energy than our pancreatic exocrine cell, and so it's putting more membranes towards the mitochondria. Okay, and again, you can see that if we look at from a cell membrane perspective, some of these smaller vesicles are going to be a smaller percentage, even smaller than our plasma membrane. So the take home from that table really is that intercellular lipid bilayers comprise the major fraction compared to the plasma membrane, right? So we want to keep in mind that even though we think about the plasma membrane, as being the membrane of the cell, there are so many membranes that are inside the cell, and with that, we need to be able to get proteins to those different locations also. So next we're going to look at how did we get from a prokaryotic cell to a eukaryotic cell. So the theory that looks to explain this process is known as the endosymbiotic theory. And for different organelles, the theory explains how these different organelles arise. So we're going to look at how the endoplasmic reticulum arrived, and also the mitochondria. So these are have figures in your textbook. And we'll also briefly do a narrative on chloroplasts. And so we want to keep in mind that with prokaryotic cells, these are similar to bacteria. They lack organelles. So they have their plasma membrane, but many of these functions are done along the membrane. So things like generating energy and such. Okay, for eukaryotic cells, we end up having organelles, and we want to keep in mind that there's this intermediate, right? So we have a prokaryotic cell, but we didn't all of a sudden jump to a eukaryotic cell. There are going to be intermediates or pre-eukaryotic cells in that process too. Okay, so with the endosymbiotic theory, what is believed to have happened is that part of the membrane actually started to invaginate in. So basically, the cell kind of turned in on itself. With that membrane had some membrane-bound ribosomes, and that enclosed around the DNA. So keep in mind that prokaryotic cells have circular chromosome that actually condenses down on itself, and so keeps itself compacted into an area within inside the prokaryotic, the bacterial cell. Okay, so during this process, where we had this invagination of the membrane, that became enclosed in around that aggregated DNA and developed into a nucleus. Okay, and then because we had ribosomes bound to this plasma membrane, it then becomes the endoplasmic reticulum. Okay, so at that point, we have this pre-eukaryotic cell, which is then in close proximity to this prokaryotic cell that's able to undergo aerobic respiration, which is able to produce energy. Okay, that cell then gets engulfed into this pre-eukaryotic cell, and because a bacteria is able to reproduce on its own, now that it's internalized, it's actually able to replicate, and this is what ends up becoming the mitochondria. Okay, a similar phenomenon happens for chloroplasts, where bacteria that are photosynthetic are internalized and then are able to self-replicate, and they become chloroplasts because they can undergo photosynthesis. Okay, so obviously we didn't all, our early eukaryotic ancestors didn't all engulf photosynthetic bacteria, or we would all be able to be photosynthetic. So it would be those early precursors to plants that did that. So some of the evidence for the endosymbiotic theory is if we actually look at mitochondrial DNA and compare that to prokaryotic bacterial DNA and look at those genomes, there's a high homology between them. And so this high homology is not likely to happen unless they actually derived from each other, so that they have like a common ancestor. And so obviously our current prokaryotic cells have evolved over time. I mean, we can just see in the last decades how they've evolved to have drug resistance, so antibiotic resistance. And so, obviously over millions of years, they're also going to evolve. And so their genomes wouldn't be identical, but the homology is so high that the evidence supports that they originated from the same area. Also, if we look at mitochondria, their ribosomes are the same size as with a prokaryotic cell, a bacteria. So the ribosomes in our human eukaryotic cell out in our cytoplasm or attached to our ER are a different size. And again, so the size of ribosomes in the mitochondria are the same as bacteria. Also, genes that are found in the mitochondria, as I mentioned, like they're the same genes for cellular respiration that a bacteria would have. So the mitochondria really becomes a self-sufficient organelle, and it can replicate on its own. Similar with the chloroplast, if we look at the genome inside a chloroplast, that genome has high homology with photosynthetic bacteria, the scion bacteria. And so again, this high homology is not very likely to have happened randomly. They would have had to have a common ancestor in order for that to happen. Also, the ribosomes inside of chloroplast are the same size as ribosomes in cyanobacteria. And again, chloroplasts have their own genome, their own within that genome, the enzymes in which they need in order to support their function. And so both mitochondria and chloroplast really are these kind of self-sufficient organelles. And it speaks to them, this endosymbiotic theory holding up. Okay. If you have any questions about the endosymbiotic theory, let me know. I find it very interesting from a microbiology standpoint. The other evidence is that there are bacteria that when our human cells take them up, they're actually able to replicate inside. So like rickettsias are an example of a nowadays bacteria that our immune cells can engulf, but they actually can survive inside of our immune cells and they actually can replicate. So the numbers will increase. So like you can watch it, them grow and divide under a microscope of an infected cell. And so again, this is, this sometimes it's called like a nesting symbiotic relationship where you have an organism inside of an organism. We can see it, we can have examples of it nowadays. And so millions of years ago, it just allowed us to go, not us personally, but it allowed those prokaryotic cells to be moved towards a eukaryotic cell. It gave those eukaryotic cells an added fitness, an added benefit, because they had these organelles that were sole function was to make energy or to undergo photosynthesis. So it wasn't that the whole cell was not preoccupied, but the whole cell wasn't being utilized for that function, just part of that. So that gave that cell an added benefit, right? So prokaryotics are limited in their size because they have to perform all these functions. And with a eukaryotic cell, it can be much larger because it can have these compartments, these organelles that provide those special functions for it. So it would give it an added benefit, added fitness. Okay. So the bulk of the rest of this lecture will look at proteins and how they are sorted, how they move to different locations. Okay. And this is, proteins have to move between compartments in 3 different ways. And so for this lecture, we're going to focus on the first 2 ways. So what's known as gated transport. So this is when the protein's produced in the cytosol, and then they would move to the nucleus. And then we're also going to look at transmembrane transport. So this is where they're being produced in the cytosol and then moved to the ER, the lumen of the ER. So we looked at the lumen of the ER with that paper for the Tech Talk, right? So we should know how they know to go to the lumen of the ER, right? KDL. And then lastly, we're going to look at transport through vesicles. And so this is where we have them moving from the ER lumen to the Golgi lumen, and then potentially exocytosis. So when we're looking at how proteins are sorted, we can think about these different pathways. So this figure does a nice job of kind of summarizing up some of these pathways, and where if it's nicely color-coded, right? So if we're talking about gated transport, proteins are going to be produced in the cytosol and then they're moving to the nucleus. If we're talking about transmembrane protein, this is coded in the blue, so they're going to be produced in the cytosol and then they're moving to like mitochondria, endoplasmic reticulum, perisomes. Where with vascular transport, this is where they have been produced in the cytosol, moved to the ER, but for vesicle transport, we're now moving to the ER to the Golgi, and then from the Golgi, depending upon where the protein is supposed to move to. So there are some signaling sequences that direct proteins to the correct compartment and where they need to go. And so I had already mentioned KDL. So KDL is where it's going to be told to go to the ER, okay, where we can have other sequences that are going to indicate that they need to move to perisome or mitochondria, or to the imported or exported from the nucleus. Okay, so I don't expect you to know all these different signals. I do expect you to know KDL. Right, and so how do we know that these signaling sequences are? So how do we know that these signaling sequences actually code for location? What we can do is transfection assays, where we can take a protein of interest. So maybe we have a protein that's normally found in the ER. We can look at its sequence, but we can know that the relevance of that sequence, because if we remove that sequence, it's going to no longer go to the ER. It's going to go into like the cytosol. We could insert in a sequence that tells it to go to the nucleus, right, or the mitochondria. And so we can change the location which it goes by changing that signaling sequence. So what we're seeing in this figure is where in panel A, normally we've got this ER protein, which is shown in blue. It's got the endoplasmic reticulum signaling sequence like KDL. And so what we've done is we've removed that signaling sequence. We've actually added that onto a cytosolic protein. We add that KDL on, and we transfect the cell with our new genes of interest. And maybe we've even coupled this with a GFP protein or something. And when we go to look for our protein, our cytosolic protein, because it has the ER signal sequence of KDL, it now moves into the ER, where because our ER protein no longer has the signal sequence, it's now found in the cytosol. So we can see how studies have been done where you can remove that and it changes the expression pattern. So I had mentioned in the tech talk, I looked at an ER protein, GRP78, and it has a KDL on its end. And so when I was doing my postdoc, I was also looking at GRP78 in prostate cancer. And so working with one of the other postdocs in the lab, he was really great at making different vectors, so you could do transfections. And so we had different forms of GRP78 that some had the KDL and other ones had no KDL. So we could look at the changes in the trafficking of the protein and basically induce cells, prostate cancer cells, into becoming more aggressive forms of prostate cancer cells if the GRP70 went to the surface where it normally isn't found. So that's my fun research story. So we're going to look next at the gated transport. So that movement of proteins between the cytoplasm to the nucleus. So keep in mind with the nuclear envelope, that is going to include the inner and outer nuclear membrane. And attached to this is also the ER membrane. And so keep in mind that it's not that we can just tweeze them completely apart. Within the nucleus, we're going to have these nuclear pores, these pores in which material can enter and exit. Okay. So nuclear pores are going to be these large complex structures that are multi-proteins together, right? So if we remember back to that photobleaching, remember how the nucleus is slower at replacing any of these, and so these proteins are going to be found in the nuclear envelope making up the nuclear pore are highly complex in multiple proteins, and so to get the replacement of all of those proteins is going to take time, right? So this image is just, these images here on this slide just do a nice job of showing you that complexity to create that pore, the nuclear pore. So it's a number of different proteins coming together, and we have, you know, both our cartoon kind of representation of it. I love transmission electron microscopy, so in undergrad I did work with an electron microscope looking at gap junctions. It's not nuclear pores, but and so I always love micrographs using electron micrograph or microscope. And so in panel C we can see that nuclear pore, so we've actually cut the membrane, and so we can see those holes, those pores, and that complex goes throughout. There's actually these cytosolic fibers in the nuclear fibrils. So just like the transport of molecules across the plasma membrane can be active or passive, and it really depends upon what molecules being transported across, the same is true with the nuclear pore complex. Depending upon the size of the molecule, it may diffuse through freely, where larger molecules would actually have to be actively transported into the nucleus. So there are conserved sequences that allow for nuclear targeting, so that it would be transported in. So in this figure here, we see in panel A, this is the normal sequence that would be found on this protein, and that allows it to go into the nucleus. And so if we look at the staining, we can see that we have this really nice circular staining present there. However, if we do a mutation, if we point mutate in this targeted sequence, we can see that now there's a change in the expression, and we actually have this protein T antigen present throughout the sides of the cell. So it is important to note with nuclear localization signals, it can be located anywhere in the protein. So KDL for the ER is found on the C terminus right at the end. With nuclear localizations, they can be embedded within the protein. And if a protein is complexing in the cytosol, if one of the proteins within the complex has the nuclear targeting signaling, it's going to move the whole complex in. So large molecules can be transported across the nuclear pore without having to unfold them. So here we're looking in this electron micrograph. If you look at where the arrows are pointing, these would be larger molecules that are being transported in. So they kind of get caught by those fibrins, and then they move through the pore. And then on the other side, we also have this fibrin or this basket that they get caught into. Okay, so export works very similar, but just kind of in reverse. And again, that would also require targeting sequence for the export out of the nucleus. So, you know, ribosomes can be synthesized within the nucleus and then exported intact through the nuclear pores. So there is a little bit like a flexibility in the nuclear pore. In order to bring proteins that contain a nuclear import sequence, it would bind to a nuclear import receptor. That receptor recognizes that localizing sequence and then helps to traffic that into the nuclear pore complex. Okay. In order to generate the energy required in order to do this, GTPase is responsible for the energy production. So GDP becomes GTP, so similar to ATP synthase, but GTPase is going to be the protein that is going to allow for energy production in order to allow for import and export of nuclear transport. Okay, so next we're going to look at, so for the next part we're going to be looking at protein targeting in the endoplasmic reticulum, so in the ER. Okay, so our blue pathway here. So the important thing to keep in mind with protein targeting for the ER that it's a co-localization process. So it's not just the polypeptide chain that is moving, but the ribosome and the messenger RNA, the RNA is also moving with it, so that as the protein is being synthesized, it can be produced into the ER. Okay, so keep in mind these proteins haven't been folded yet, so there's no requirement to unfold them. And so just keep in mind that for other organelles like the nucleus, all the proteins need to be made before translocation transport occurs. So for proteins that are being moved to the ER, for this way they're actually being produced and synthesized as they are moving, translocating. So keep in mind that the endoplasmic reticulum has two different functional domains, so the rough ER and the smooth ER. So the rough ER is going to be embedded with ribosomes, so ribosomes are going to be docked on it, where smooth ER is where more enzymes are going to be located, and so these are important for lipoproteins synthesis and detoxification that occurs. But the ER in itself is a pool of intracellular calcium. So there's a large amount of calcium within the ER, which allows for proteins to assist on folding and such. So if we were to actually take the ER and try to isolate it, it can actually create its own little microsomes. So if you remember, like with the liposomes, so we could take phospholipids and create these little beads with them. We can do something similar with the ER. And so characteristically, they are going to actually be between 100 to 200 nanometers in size, and that the smooth microsomes that would end up being formed from the smooth ER would actually have a lower density. So the rough ER are going to have a higher density, and that's because of the ribosomes attached to them. They're going to end up having more mass to them. So looking at an overview of protein translocation across the ER membrane, we can see that we will have our ribosomes attach the message RNA, and we can have multiple ribosomes attached at different parts of the message RNA. And so as that polypeptide chain is being produced and that there's a recognition of this signaling sequence, that will traffic it to our channel, our translocator, that allows for that polypeptide chain to continue to grow. And as it's growing, it's actually inside the lumen of the ER. Okay, so a couple added pieces to this is that there's going to actually be a signal recognition particle that is going to bind to that signaling sequence, and it will bring that ribosome and the message RNA to the ER, where it will bind to a receptor. So our signal recognition particle will bind, or SRP will bind to a receptor that's on the surface of the ER, And that puts the ribosome in the right position so that the polypeptide chain can enter in that channel, that translocator. So you might remember back to the tech talk where with the ER membrane, they were looking at Ch15, and so that is a channel that allows for that polypeptide chain to enter into the ER. So what we're looking at in blue here is what we saw in the tech talk in that second figure number 2, which was actually the first figure we looked at, that was in the green, the emerald color. Okay. So once the polypeptide chain has started to enter in, a signal peptidase, this enzyme will actually cleave with the signaling peptide and allow for that when synthesis is finished, when the whole messenger RNA has been read and amino acids have been added, that mature polypeptide chain is now in the lumen of the ER. Now, some of the ER signaling sequences do not get removed, so KDL is one that does not get cleaved off because KDL allows for retention into the ER lumen. So if you don't have the KDL, you're going to move on in your journey. So this is just showing you what that signal recognition particle looks like, or SRP. So this is going to become part of the ribosome protein complex. So it's going to bind to that signal. You can see how there's a sequence binding pocket. So it's going to bind to that signal that says bring me to the ER, but it's also going to anchor onto the ribosome, and it's stabilized. Then it's going to bind to its receptor, and all that stabilizes this complex so that the ribosome can dock. So as you can see in this figure, here we have all the pieces coming together. We have that signaling sequence that's coming off of the newly synthesized polypeptide chain. We have our SRP, our signal recognition particle, binding on, kind of reaching over and stabilizing that interaction. And then we have a receptor binding site that's now available to bind to the receptor that is on the surface of the ER. Nearby it is the protein translocator, or the channel that's going to allow the polypeptide chain to come into place. Once that interaction has been stabilized and the polypeptide chain is entered into the lumen of the ER, this can allow dissociation between the SRP and its receptor so that it can be utilized to bring the next ribosome and polypeptide chain in. So it is a common pool of ribosomes that are found both in the cytosol and the ER, so we can think of these as being dynamic, and they are going to, your cytosolic ribosome is going to bind to your messenger RNA and then move to the ER. So it's not that the ribosomes that we see on the surface of the ER are there for their whole entire life. They would disassociate, new ones are going to come in, and because proteins are always being synthesized within a cell, unless there's some kind of drug that is the cell is being treated with some kind of drug that stops protein synthesis, like doxycycline. It's an antibiotic. So unless we've stopped protein synthesis from occurring, we're constantly going to be having new ribosomes coming in, docking in the rough ER, and then dissociating when they're done so that they can go through this process. Now, not all ribosomes, again, are going to end up docking to the ER because some of them might just need to synthesize proteins that can be free in the cytosol. Okay, so again, keep in mind that this is a really dynamic movement that's occurring for the ribosomes. So here is an image of Ch15. This is that translocator, the channel, which the polypeptide chains are going to move through. So again, this is what we saw in the tech talk in that emerald probe, with that probe. Okay. And so we can see how there's this channel that's present, and so we can imagine something could move through that channel. So when we have that signaling peptide present, it actually will open up the channel, so it's easier for the polypeptide chain to enter in. It causes this kind of plug, so you can see that yellow strand coming up. That actually kind of moves down out of the way, and so your polypeptide chain from your ribosome can then move in. So this is just giving you another view. So in the blue is where we have our Ch15, our channel. And so it's actually they complex, so there's 3 of them together. This allows for there to be ribosome attachment sites. So similar to, you know, the aquaporin, which had that 4 subunits and allows for water to go through, right? It was 4 subunits. So this one's also made up of 4 subunits. And so we can see how we have our ribosome docking, and so we have to have attachment points there also. So signaling peptidase is going to actually cleave off as the ER signaling from proteins that are not going to stay in the ER. So they just needed the signaling sequence to get them to the ER, but they're going to move on in their journey, okay? So the amino acids that are found in that signaling sequence can be recycled. So again, we can just think of this sequence as kind of a start transfer sequence, okay? So we can also have a stop transfer sequence. This is actually what's going to end up becoming the transmembrane domain. And so again, there'd be a sequence in there that when it's recognized by the translocator protein, it knows to shift it, don't keep moving it down. It actually needs to go into the membrane. And so then it would be embedded into the membrane for that. Now, if we have our start transfer sequence internalized, this would give us a different outcome than if it's at the beginning. So the location of these stops, of the starts and stops, affects where the protein would then be located. So if the start transfer is at the beginning, like we previously saw, we would have it move into the lumen. But if the start transfer sequence is internal, we're going to get the recognition by the signal recognition particle. It's going to move to the translocator, and then it would become a transmembrane section of it. So different strategy than with our stop transfer. So with the stop transfer, again, we move that into location into the transmembrane, but with a start transfer that's internalized, it would end up moving into the transmembrane, and it just gives us a different orientation than what we would see in the other case. Now, if we had a start transfer and a stop transfer sequence, depending upon locations, right, we could end up with a 2-pass transmembrane protein, right? So the more these start transfer, stop transfers, the more transmembrane loops we're going to end up having, okay? And so in this case, we have our start transfers internalized, so this means it's not going to move out to the lumen of the ER. It's going to become transmembrane, and then we also have this other stop transfer, so again, that's going to be transmembrane, and so we can see how we have these 2 transmembrane sections present. So the last piece with this lecture is looking at constellation. So most ER proteins are going to be cosylated. So constellation occurs on the asparagine, And so oligosaccharide transferase will transfer these sugars from phospholipids onto the proteins. Okay, so I'm actually going to go to the next slide. So you might remember from our chapter 4 lecture, we touched upon the idea that we have this glycocalyx that's present outside the cell membrane, and that's made up of constellations. And so if we think about when the membranes are being made, they're going to end up being part of the plasma membrane. Constellations allow for kind of an orientation and a marking of these proteins. And so the constellated sides potentially are what's going to become our extracellular portion of the protein, right? Because what's in the lumen of the ER, as it moves through transport, those membranes would end up becoming the extracellular portion of the plasma membrane. Okay. So constellations are also, these oligosaccharides are also a way to mark the proteins to determine if they're folded correctly, right? So calnexin is a chaperone protein, and it has a lectin binding domain, and so it allows for these proteins to bind to calnexin, and calnexin is able to assist in the folding of the protein, so that it would be folded normally, right? And so we can see how this protein, calnexin, is going to help to fold it, and it's using that constellation as a way in order to stabilize and to make sure that it's holding it correctly so that it could fold correctly. And so then once it becomes folded, it can then exit from the ER if it's not being retained there. So proteins that are misfolded would be exported to the cytosol. So this process is done by ubiquination. So that tells the protein to go to the proteasome, and the proteasome is a structure that degrades. So proteins go in, they get digested, degraded, and so then those amino acids can be recycled. So again, this idea of post-translational modifications that can occur to the proteins that act as reporters to tell whether things have been folded properly or not is useful. The textbook briefly has a figure about the unfolded protein response. So if there's proteins that are not being folded correctly, there is a stress response that happens in which additional molecular chaperones are produced. And so it's an important response they have, and that was part of my undergraduate work, was looking at the unfolded protein response, and GRP70 plays a role in that. So another modification that can happen is that proteins in the ER can be anchored through GPI. So with GPI proteins, this can allow them to be anchored so they can move through to secretion, and then through phospholipidase activity, the GPI can be cleaved and allows for the protein to go on secreted, but it's not that it allows the protein to move through this secretion process in a controlled fashion. And GPI anchored proteins can assemble into lipid rafts. So it's a way to, again, regulate how proteins are moving through the process. Okay, so this concludes the topics for Chapter 15 related to Part 1 and Part 2. There is another audio lecture for the third part, where that involves the movement from the ER to plasma membrane. and other structures. Okay, so for the complete audio lecture for Chapter 15, you do have to listen to both audio lectures. So thank you.